Hi, my name is Brett Hale, and I am a PhD student in the lab of Dr. Isela Wijratna at the Arkansas Biosciences Institute in Jonesboro, Arkansas, which is on the campus of Arkansas State University. Our lab studies soybean, which is an important row crop in the state of Arkansas, and we are really interested in interactions between soybean and microorganisms that live in the growing environment. And in studying these interactions, we really focus on diseases of soybean. So one of the most prevalent diseases in the state of Arkansas is called sudden death syndrome. It's caused by a fungus, Fusarium virguliforme. And we like to study interactions between soybean and Fusarium virguliforme to see if we can better understand what interactions are taking place and how they are influencing the native microbiome of the plant. And in doing so, hopefully we can develop more environmentally benign methods to control the disease. So what we're doing today is trying to look at microbiome structure and then function at a later time point to understand how the pathogen suppresses the soybean rhizospheric microbiome, and then how soybean plants that are tolerant and can fight off the pathogen, how their microbiomes differ from those that are susceptible. So what we're doing today is using a technology called Oxford Nanopore 16S barcoding and sequencing to look at the microbiome structure following Fusarium virguliforme infection in soybean. So Oxford Nanopore has several advantages over other methods of amplicon sequencing to look at the microbiome. So one of the major reasons that we're interested in Nanopore is because it leverages long-read sequencing, which allows us to look at multiple hypervariable regions in a marker gene. So when we want to look at bacteria or fungi or other eukaryotes or prokaryotes that comprise a microbiome, we use marker genes that allow us to amplify that gene for all microbes that are in a biological sample, and then we leverage hypervariable regions to classify taxonomy. And this is a really good approach to get a zoomed out view of what's going on in the microbiome. However, those hypervariable regions are oftentimes conserved between taxonomically related microorganisms. So by leveraging long-read technologies such as Oxford Nanopore, the hope is that we can then get better taxonomic resolution so that we can look to see what's happening to the microbiome at the genus and potentially species level. So today is just going to be a walkthrough of what that looks like. So we had, before this video, isolated DNA from soil samples of soybean plants that have been infected with Fusarium virguliforme or treated with a sterile solution. And then those came from the rhizosphere of soybean plants that are known to have genetic tolerance to the pathogen or to be susceptible. So we isolated the DNA. It had been quantified and purity checked by our lab. And we start today by barcoding these samples so that we can individually give them a label or a classifier. So we amplify that 16S gene. It's barcoded with an individual tag per sample. And then we're going to purify that, get just that amplified tag gene, combine our samples, and then run that on an Oxford Nanopore MinION sequencer. And that's what we're going to show you